All right, so here we're going to talk about glycogenolysis, or the breakdown of glycogen, and either the use of the glucose or the release of the glucose to maintain blood glucose levels. All right, so liver glycogen is going to have a source of blood glucose. And so we again have our glycogen molecule. And glycogen molecules are going to be converted to glucose 1-phosphate. And the glucose 1-phosphate will be converted to glucose 6-phosphate. And depending if you are in the muscle, you're going to use this glucose 6-phosphate for energy. Or if you are in the liver, you are going to have glucose 6-phosphatase. And you are going to release free glucose to maintain blood glucose. So glucose 6-phosphatase is very important, and it is what's going to remove that phosphate, which enables the glucose molecule to leave the liver cell, the hepatocyte, and enter the blood. Now, glycogen degradation needs different enzymes than those that were building it, so you simply can't use the same enzymes. The enzyme that's going to do the most degrading is glycogen phosphorylase, and this is going to remove glycosyl units 1 unit at a time from the non-reducing ends. So synthase would build at the non-reducing ends, and glycogen phosphorylase breaks down at the non-reducing ends. And so you have, again, you have these branches, so you have multiple non-reducing ends, and your glycogen phosphorylase can clip these off, and you're going to get glucose units. So here it's showing that you can get 8 clipped off, and you can make 8 glucose 1-phosphates. Now, as you get closer to these branching points, your glycogen phosphorylase is not able to fit. So it becomes a little too crowded. And so you're going to have a debrancher enzyme that removes the glycosyl units near each branching point. So let's talk about these enzymes. We have 3 key enzymes for glycogen degradation. The first is glycogen phosphorylase. And glycogen phosphorylase, again, is going to work on the non-reducing ends. It removes 1 residue at a time, and it repeats until it reaches the 4th glucose unit from a branching point. So glycogen phosphorylase will release glucose 1-phosphate. And then the glycogen molecule is actually shortened by one residue at a time. This is going to have the ability to preserve some energy in that glucose 1-phosphate bond. The next enzyme is our debranching enzyme. Now, our debranching enzyme has 2 functions. It's an oligo alpha-1,6 to alpha-1,4 glucan transferase. And so once we get to this crowded branching point shown here, you're going to have the first reaction occur on the debranching enzyme. The first reaction is going to be a 4-to-4 transferase. And so remember, we have our C4 here, and we have our C4 here. And so you're going to get your sugar molecules attached at that. Your branching point is going to move and attach to the end of your glycogen core. Now, after this happens, you still have 1 glycosyl unit left, because that portion of the transferase cannot remove that 1 glycosyl unit. So now you need the second reaction of the debranching enzyme, which is an alpha-1,6 glucosidase reaction. And in this case, it's going to remove a single glucose. Now, this glucose is not phosphorylated, and so this glucose can be released immediately or can be used. So it forms a free glucose. And then you have a long glycogen core, and then your glycogen phosphorylase can start degrading or start removing the glycosyl units one at a time again. Now the last enzyme is this phosphoglucomutase. And the phosphoglucomutase is going to take the glucose 1-phosphate, and you're going to have reorientation, and you're going to generate a glucose 1,6-bisphosphate. And so here we have our phosphate on our anomeric carbon. And so in this case, we're going to add a phosphate to our carbon 6. And then your carbon 1 phosphate is going to be released, and you're going to generate glucose 6-phosphate. So new phosphate has to come in to bind to carbon 6, and then the phosphate on carbon 1 will be released. Now overall, this is a picture from your book about the synthesis and degradation of your glycogen. So again, we always start with glucose. Glucose gets picked up. Your glucose is going to react with hexokinase or glucokinase and form glucose 6-phosphate. Now, of course, depending on what the status of the cell is, glucose 6-phosphate has many pathways to go to. However, if it is in a fed state and you have a lot of glucose around, your glucose will want to be stored as glycogen. And so glucose 6-phosphate through phosphoglucomutase becomes glucose 1-phosphate. Phosphoglucomutase is the same enzyme that can go back and forth between glucose 6-phosphate and glucose 1-phosphate. You then have your charged glucose, your activated glucose, UDP glucose, with glucose pyrophosphorylase. And this UDP glucose also can go to other pathways. As you continue on to make your glycogen, you have your glycogen primer, which is, of course, going to be your glycogenin, which is typically in every cell already there, and your non-reducing ends are already present, and you're going to build on those non-reducing ends. And so you have your glycogen synthase as well as your branching enzyme, and these will build your glycogen molecule. Now on the other side, for glycogen degradation, of course, you're going to have your Debrancher enzyme, and you're going to have a free glucose. And then you're also going to have your glycogen phosphorylase, which will remove each glycosyl unit one at a time and generate glucose 1-phosphate. Glucose 1-phosphate can then go to glucose 6-phosphate, and you have the glucose 6-phosphatase in your liver, and this will release a phosphate, and you can release glucose. Now in the muscle, that glucose 6-phosphate or any other cell in your body obviously would use that for energy. Just a little bit on the glucose 6-phosphatase. Glucose 6-phosphatase is actually in the endoplasmic reticulum. It's embedded in the membrane. The active site is facing the lumen of the endoplasmic reticulum. And so there is a glucose 6-phosphate transporter that transports glucose 6-phosphate into the lumen of the endoplasmic reticulum. In the endoplasmic reticulum, you're going to get release of a phosphate, inorganic phosphate. There is an inorganic phosphate transporter to transport that phosphate out of the lumen of the endoplasmic reticulum. There is also glucose transporter T2 on the surface of the endoplasmic reticulum in your liver cells, as well as some kidney cells, and you release that free glucose through GLUT2, and then this increases the blood glucose concentration. And of course, muscle will use glycogen stores for energy, and liver uses it to maintain blood glucose levels. So the breakdown of glycogen or glycogenolysis is the degradation of your glycogen molecules. You're going to have two different enzymes that are going to be important for doing this. You're going to have your glycogen phosphorylase, and your glycogen phosphorylase is going to release glucose 1-phosphate. You're also going to have a debranching enzyme hydrolyzing the alpha-1,6 linkages and also releases a single free glucose. It has two active sites to do this. Use of glucose by degrading your glycogen stores is going to be different for liver, which maintains blood glucose, and muscle cells, which needs it for energy.